Hello everyone, welcome to today's live broadcast, Melanoma, View from the Frontlines of Immunotherapy and Precision Medicine. I'm Christina Jewell of LabRoots and I'll be your moderator for today's event. Today's webcast is presented by labroots.com, the leading social media site for science professionals. Before we start, there are a few instructions. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. Answers are welcome too. You can do this by hitting the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can and we'll follow up if we don't have time today. Want a better look? You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or use the Q&A button. We'll make sure we try to resolve any issues. Now let's get right to today's presenter. We are proud to welcome Louise Perkins, PhD, Chief Science Officer at the Melanoma Research Alliance.
Louise Perkins, I'm the Chief Science Officer at the Melanoma Research Alliance. My pleasure to join you.
Dr. Perkins is responsible for the development and implementation of the MRA's scientific strategy. Her interests center on translational research, including genomics, drug discovery, and advancement of novel therapeutic approaches. Dr. Perkins became a member of the board of directors of the Foundation of Sarcoidosis Research in 2015. Prior to joining the MRA, she served as Chief Scientific Officer at the Multiple Myeloma Research Foundation for 5 years, where she led the expansion of its venture philanthropy activities, including the Biotech Investment Award Program and the development of the scientific direction of its Compass longitudinal study. Previously, as the Director of Cancer Research at Bayer Pharmaceuticals, Dr. Perkins contributed to advancing novel targeted therapies toward clinical study. And at the Schering Plough Research Institute, she led a cancer research group participating in early-stage programs. Her complete bio is found on the LabRoots Welcome. Please join me in welcoming Dr. Louise Perkins. Dr. Perkins?
Hello, everyone. This is Louise Perkins. I'm the Chief Science Officer at the Melanoma Research Alliance, and it's my pleasure to speak with you today about a subject that's very important to us at the Melanoma Research Alliance, the view from the front lines of immunotherapy and precision medicine, which is where we are in melanoma right now. As we get started, I want to just give you a little bit of history about Melanoma Research Alliance, which provides an orientation to where we are and why I think this is such an important subject. The Melanoma Research Alliance was founded in 2007 by Deborah and Leon Black, shortly after Deborah's diagnosis with melanoma. Her care has come together very well, but she was challenged at that point in time to establish in melanoma a foundation committed to the elimination of melanoma suffering and death, very much in the image of the Prostate Cancer Foundation, and we were created under the auspices of the Milken Institute in order to achieve that mission. So since that time, MRA has been able to become the largest private funder of melanoma research. We have awarded over $79 million to researchers around the world, all in academic institutions, and we have an expectation this year, coming up in 2017, to award at least $7 million more for research covering the gamut of established investigator science, early career investigators, pilot awards for early emerging things with a strong hypothesis, as well as for industry partnership programs. Likewise, one other key feature of our program is that we are very much committed to interdisciplinary team science, and although we are not funding it this year, we have supported nearly 2/3 of the dollars that we've provided for this kind of interdisciplinary work. just wrapping up on MRA, our focus is on translational research. We have awarded individuals and teams, as I said, and about 85%, pushing 90 actually, is the fraction of our dollars that have gone to treatment-related science with the balance in prevention and new diagnostics. So that's a history of MRA, and you can see that we're really passionate about the field, about working towards our mission with all of the stakeholders in the business. So with that, then I'm going to just push on and give you a brief introduction to the agenda for the session. And once I get through this, I'll ask my colleagues to push out the polls. So what we'll cover is an overview of melanoma. And the biology of melanoma actually provides the basis for the treatments that have emerged and also provides hints as to where we can go in the future. I'll cover some of those treatments and really the fantastic progress that has happened to date, from immunotherapy to precision medicine, targeted therapy, and then just wrap up very quickly with some comments on the future and where I think things will be going for the field. So if we could have the polls go out, that would be great. So let me move on to the melanoma overview. I'm going to cover a little bit about the facts and figures of the disease, some of the biology, and then touch on really some of the key firsts that melanoma finds itself in. So first of all, you should see that melanoma incidence is rising. The statistics shown on the right-hand part of the slide illustrate the increase in melanoma new diagnoses per 100,000 people. This is just U.S. data, and I just pulled it down from the SEER website recently. You can see there's been a steady increase, in fact, a tripling of new diagnoses for melanoma. And that lower flat line that really doesn't change shows that deaths have pretty much remained flat. In fact, in the U.S. this year, there are approximately 76,000 new diagnoses expected and 10,000 deaths, unfortunately. The median diagnosis for melanoma is at about 62 years of age, and it is the most common cancer diagnosed in 25 to 29-year-olds. Some people hypothesize that this has something to do with the use of tanning beds, sun exposure, that sort of thing, and almost certainly those things do contribute to the onset of melanoma, as you'll see in the genesis of melanoma due to genomic changes in a few slides. You can tell by this great delta between the new diagnosis and the death rate that clearly melanoma, when it's caught early, can be managed very well, and surgery is a mainstay of that management. However, if it's caught in the advanced stages, when it's spread to distal organs, there is a poor 5-year survival of about 15%. So the overview for melanoma and its incidence in various populations demonstrates that melanoma has the highest incidence in fair-skinned individuals. If you look at the top of this slide, you'll see that for the white race that there are the largest number of cases of melanoma. The fact is that melanoma is more common in males than in females. But if you look at other individuals with darker complexions, melanoma indeed is present and diagnosed at a measurable rate. In fact, I can think of many individuals who have unfortunately been diagnosed who have darker complexions. And so it just points out that melanoma can affect anyone, but indeed the vast majority of melanoma patients are fair-skinned individuals like myself who have a propensity to the disease for a variety of reasons. And a little bit about how melanoma presents in the clinic is shown in this slide. You can see beginning at the top left and walking from panels A through E that cutaneous melanoma occurs in multiple sites with different presentations. At the top left in panel A, superficial spreading melanoma is the most common presentation of melanoma in the clinic. Panel B shows a melanoma that actually is amelanotic. In other words, it's lost its expression of melanin, the dark pigment, and it is that melanin pigment which is the pigment that protects us from sun damage. Panel C shows a nodular melanoma, and listening to the dermatologists who are participants on our various scientific advisory panels leads me to believe that these nodular melanomas are difficult to diagnose and dangerous when they're diagnosed. So these are a particular a challenge. And panel D, acral lentiginous melanoma. Acral melanoma occurs on the palms of the hands, soles of the feet, and under the fingernails. This is the kind of melanoma that actually is most frequently presented in patients with darker complexions. And finally, ocular melanoma, this particular one in the back of the eye. So these are some of the presentations of melanoma. And this next slide on melanoma biology shows the genesis of the disease. Given the size of this particular illustration, I'm not going to be able to go through it in a great deal of detail. I do want to point out to you that as you can see kind of in the middle of this panel, beginning with stage 0 and stage 1 at the bottom, that melanoma forms in the pigment-producing cells of the skin, the melanocyte. That dark splotch in the middle of the panel above the stage 0 text would be the earliest presentation of melanoma. But as with many tumors, as it grows larger and invades more deeply into the surrounding tissue, it becomes increasingly dangerous. And you can see as you progress to the far right that these deep, thick melanomas are the ones that are the stage 4 melanomas that are particularly vexing in the clinic and are the most dangerous of the melanomas. Now, what drives the formation of melanoma, and how does this help us as new treatments have been discovered over time? Well, what's been observed in sequencing studies, and there are many of them, including the TCGA studies and work that's been done at Yale and other centers, studies that we ourselves have supported with TGen leading the genomics work, shows that melanoma is in fact the cancer with the highest mutational rate of any cancer that's been sequenced to date. In this particular chart, you can see on the logarithmic scale on the y-axis and highlighted at the far right that the red box surrounding the melanoma samples illustrates that melanoma has the largest number of mutations per megabase of all of the cancers analyzed. And if you look at the bottom of that red box region, you'll see the block of yellow color at the bottom. That block of yellow color illustrates the kinds of mutations that are observed in melanoma. There is a predominance of C to T transitions observed in melanoma, which are a hallmark of UV-induced damage. So that signature of UV exposure really provides us, along with the information that there are many, many mutations seen across the melanoma tumor genomes, that there is in fact a mutational event that contributes to the driving of this disease, and also helps us to at least think about and provides food for thought as to why melanoma may in fact be the most immunogenic of the cancers and may be particularly responsive to treatments that re-engage the immune system in the treatment of melanoma. So I'll come back to that point about the role of mutations in the production of the new antigens, so-called neoantigens, in a moment. Just spending a second to talk about melanoma targets specifically, what we know from the analysis of melanoma genomes, looking at the whole exome sequencing, WES, that the signaling through the RAS-MEK-ERK pathway is clearly dysregulated in melanoma. At the top of this chart, in work from Michael Krauthammer at Yale, you can see that in this set of samples, there were about 40% of the melanoma tumors that were analyzed that had mutations in one of the 3 RAF proteins, BRAF. BRAF is the most frequently mutated target in melanomas, and this has been seen in a variety of samples. And indeed, the predominance of mutations there are mutations at the 600 position from a V to an E. So V600E mutations are the most common, And that's shown at that top left red block of left red block of the panel here. The rest of the mutations that are shown are in RAS. That's a predominant set of mutations. And the RAS protein that is the target for mutations actually is NRAS. The NRAS mutations that are seen are activating mutations, and those are occur at about 15 to 20% in most samples that have been analyzed across all of melanoma. I would actually point you to another resource for looking at tumor genomics and melanoma genomics in particular, and that is the CBio portal. It's cbioportal.org that can be found. It's a great tool to noodle around on tumor genomes if you have that interest. The other part that I wanted to point you to is that there are other mutations that are seen in melanomas that again confirm the role for RAS signaling through the RAS-MEK-ERK pathway. NF1, which is a GTPase activating protein, which turns off RAS signals, is also mutated and inactivated in some melanomas. And then across the rest of the genome of melanoma samples that have been analyzed, you see some usual suspects like P53, P10. You see CDKN2A mutations and also other CDK4, CDK6 activating mutations across melanomas. So that's kind of a deep dive into what drives melanoma. We know a fair bit about it, and fortunately, some targets have occurred from this information, and these targets have been driven into therapeutics. Indeed, there have been a number of firsts from melanoma based on all of this biology. And I've summarized this on this slide 11 here. Really leading the way in immunotherapy and precision medicine. This is what melanoma has seen happen since the first approvals of the first immunotherapy for any cancer, the first checkpoint drug for any cancer, I should say, with ipilimumab in 2011. And most recently, the first checkpoint therapy for the adjuvant treatment of melanoma, ipilimumab, or IPI as it's sometimes called, was approved for that by the FDA in 2015. Likewise, the first BRAF inhibitor approved for any therapy was vemurafenib, with its approval in 2011. And I should point out that the approval of IPI and VEM in 2011 were the first approvals for melanoma in over a decade, and really started this landslide of progress that we've seen in the melanoma space. There have been many other firsts for both melanoma and for other cancers that this work has enabled. The first MEK inhibitor approved in any cancer, trametinib. The first combination approval in melanoma of dabrafenib and trametinib. The first anti-PD-1 drug approved in the US, pembrolizumab, in 2014. The first approval of any PD-1 drug around the world was Nivo. It was approved earlier in Japan. First checkpoint combination therapy of ipi and nivo in any cancer in 2015. T-VEC, the first oncolytic viral therapy, was approved in melanoma. And vemurafenib and cobimetinib showed that you can, in fact, approve an agent in combination where there is no single agent approval. Cobimetinib is not approved as a single agent for melanoma in the US, only in combination with vemurafenib. So many, many firsts seen in the melanoma field, and we think that this is really just a clear demonstration of progress for the field. I'd like to bring up the results of the poll if I could. I may ask the organizers to help me with that. So the first poll question was to poll the audience's interest in the topic of the presentation today. I'm interested in this topic from the perspective of cancer patient or patient caregiver, scientist, clinician, or other. Most people on the phone, about 3/4 of you are scientists, and the rest said that you fell into the other category. The details for the second poll question, I have myself or I have a patient friend who has received immunotherapy, targeted therapy, or participated in a clinical trial. It's quite interesting. About just under half of folks have targeted therapy, say that they have or have a patient friend who's received targeted therapy, and about a third immunotherapy, and just under a quarter have participated in a clinical trial. It is not a typical population of people, I think, based on that, but I appreciate your interest in that, and hopefully you'll learn some more about this particular field and how it applies to many other cancers in the future. So moving on with the presentation, just to summarize for melanoma overall, as I mentioned, sun-exposed melanoma has the highest mutational rate of any cancer. There are prevalent mutations in the BRAF and RAS pathway. Personalized medicines have been developed that target these, including a number of drugs that I'll come back to later in the presentation. Tumor neoantigens from mutations, as well as shared antigens. I should mention, it's not just all about neoantigens. Appear to be providing targets for the immune system to react to melanoma. And there have been numerous firsts in the treatment of cancers that have emerged building from these disease features and years of research. So let's move on now to immunotherapy and melanoma. Just a little bit of background on this particular topic. It's been known for some time that melanoma is one of the most immunoresponsive cancers. Dr. Coley used infections at the turn of the century to target cancer for a variety of reasons related to unsteadiness of results and toxicities. This proved not to be a therapeutic approach with lasting legs. However, high-dose interleukin-2 was approved for melanoma by the FDA in 1998 and remains in use for melanoma, as does interferon, and interferon was approved as adjuvant therapy in 2011. So one of the earliest of the several early immunotherapies approved for melanoma. But there are significant dose-related toxicities with the use of these agents and somewhat limited activity in terms of response rate. So this has really spurred the interest in developing newer approaches. What are some of those newer approaches? This particular slide really just summarizes that some of the vast biology in the area of immunotherapy. This is a very simplified view of some of the key features that enable the working of immunotherapy in melanoma. If you look at the top left of this slide, you can see peptide or tumor vaccines as an opportunity for use to stimulate immunotherapeutic responses. In the lower left, cellular immunotherapy, another approach that's gained a lot of traction in some cancers, in particular the hematological malignancies, and is still being studied and has been studied aggressively in melanoma, but is still the center of a lot of other work. Biological response modifiers, such as the interleukins and interferon I already mentioned, shown at the lower right. But the use of monoclonal antibodies to trigger different reactions to re-engage the immune system, this has become one of the central features of melanoma treatment, and also is a very new tool that's allowing a lot of work in a lot of other areas. And why is this of interest, and how does this work? Two agents that have gained, two approaches, I should say, that have gained approval are approaches that target CTLA-4, exemplified by the approved drug ipilimumab, and anti-PD-1 drugs that have been approved are pembrolizumab and Nivolumab. How do these work? If you look at the tumor cell depicted on the right-hand side of this image, you can see the tumor cells, as part of how they go about evading the immune system, express on their surface markers that turn off activated T cells that would otherwise react to these tumors and foreign antigens or shared antigens that are recognized as being foreign. And these activated T cells would then go about and target the tumor cell and kill it. But because of the signals being presented on the tumor cell, the T cells are turned off. And what's been shown is that through the use of monoclonal antibodies like ipilimumab targeting CTLA-4 expressed on the surface of the T cell, or PD-1 using either pembro or nivo expressed on the where those antibodies target the PD-1 on the T cell surface, the disruption in signaling between the cell surface markers on the T cells and the tumor cells enables a reinvigorated immune response. And some of these responses have turned into very strong benefit for patients. In particular, in data that was presented in 2013 by Dirk Schadendorf, there actually is 10-year survival for patients treated with the CTLA-4 antibody ipilimumab, and it's actually at about 20% of patients. So if you look at the blue curve here, you can see that around about 3 years after treatment for of patients with advanced metastatic melanoma, so these are patients who would have otherwise had about 15% 5-year survival. You can see that their survival flattens out at around 1 out of 5, at around 20%. And that overall survival is durable for out to 10 years. It's really quite remarkable, and although a modest improvement over what had been expected, an amazing improvement for these patients. How do we improve on ipilimumab was the question that was addressed by the field. And the emergence of PD-1 antibodies has really been a major benefit in that. So here's the next series of slides talk a little bit about the activity of these PD-1 antibodies. For example, using pembrolizumab as one drug on this particular slide, you can see that there were 2 schedules of pembrolizumab tested, an every 3-week schedule in orange and an every 2-week schedule in blue. It's the every 3-week schedule that was actually the one that was approved by the FDA. But in both cases, what you can see in this overall survival curve out to 18 months is that treatment with the anti-PD-1 antibody did in fact improve the overall survival of these patients relative to ipilimumab alone in this relatively short time frame. This is likewise true for nivolumab alone, the PD-1 antibody nivo alone, or nivo and ipi in combination. So I'll draw your attention to the blue curve, which is nivo alone, the orange curve, which is nivo and ipi, compared to the green ipilimumab curve alone. And here, looking at progression-free survival as the endpoint, you can see that either nivo or ipi and nivo in combination appear to have a marked improvement of PFS compared to ipilimumab alone. So these drugs are actually very much changing the face of cancer care, not only for melanoma, but for other cancers. Now, combination therapy is frequently used in cancer, and we know it's very important, but often with combination therapy comes additional toxicity. And indeed, on the dose and schedule used for ipi and nivo that is demonstrating some tremendous benefit, you can see that there have been observed about 50% of patients with grade 3-4 AEs, and that compares to about 24% of patients experiencing grade 3 or 4 adverse events in patients treated with ipilimumab alone. These side effects are significant enough that about half of patients discontinue treatment. If you look at the panel on the right-hand side of this slide, you can see a swimmer's plot. The blue part are the patients who remained on treatment at the portion of time during which those patients remained on treatment. The red part, the post-treatment discontinuation time frame. The black arrows at the end of each of the lanes, an ongoing response observed. And then at the bottom, so the vast majority of this is for ipi and nivo in combination. And at the bottom, that's the comparison to ipilimumab alone. What this illustrates is that there were a lot of treatment discontinuations that took place, but interestingly, responses continued after those treatment discontinuations. And it's an important question for the field overall to explore what is the right dose, what is the right combination, what is the right schedule, what is the right duration of treatment. And kudos to the developers for pushing things ahead. Kudos to our researchers for pushing things ahead quickly to get these treatments out to patients as soon as possible. But refining all of those things is the subject of a lot of research that's going on right now. A picture's worth 1,000 words, and I wanted to just show you one of the dramatic effects that's been published in the New England Journal in a case report of a very rapid, dramatic effect of the combination of ipi and nivo on a patient. This shows at the top left the image of melanoma that was protruding from the body wall of this particular individual. You can see the photo on the top left and the CT scan on the lower left of that large mass with the star at the center of it in the middle left of that lower left panel. Just a few weeks after treatment with the combination, as you can see on the top right, this mass resolved itself and created essentially a hole in the body wall. And then on the lower right, you can see several weeks after that, a complete resolution of that mass. This is obviously a dramatic and remarkable response, and the sort of thing that drives the kind of increased interest that there is in combining multiple checkpoint inhibitors, not only with PD-1 and CTLA-4 targeting activity, but also with other drugs as they move forward. Given the side effect profile, however, one of the questions that's commonly asked is, well, how do we use these drugs in the right patients at the right time? Are they to be used for everyone? Are there biomarkers that help us to understand in whom they ought to be used better? This is a nice summary that was done by Drs. Sunshine and Taub at Johns Hopkins, looking at across a variety of clinical programs studying the anti-PD-1 antibodies on the left, Nivolumab and pembrolizumab, and anti-PD-L1 kind of sister antibodies on the right, and putting together all of this data, looking at one key question. Does PD-L1 expression, that is the ligand for PD-1, does PD-L1 expression relate to response to anti-PD-1 or anti-PD-L1 therapy? And if so, in what way? And what about PD-L1 negative patients? So I'll just focus you on the Nivolumab melanoma data, which is the second set of bars on the far left. And you can see that in the red bar, the PD-L1 positive patients have a more pronounced response than the green bar, the PD-L1 negative patients. And that general pattern, as you scan across this whole chart, seems to play itself out. PD-L1 positive patients have a more pronounced response to PD-1 or PD-L1 based therapy as compared to their PD-L1 negative counterparts. However, you will see that the PD-L1 negative patients do in fact have a measurable response. And so what this tells us is that this is a helpful biomarker in some ways, but not a perfect biomarker. And these observations have driven the field to continue to look at many, many other approaches. Pam Sharma, for example, from MD Anderson, and Suzanne Topalian from Johns Hopkins have noted that PD-L1 expression and PD-1 expression are not stable over time. They go up, they go down. And so it might be that there are other more complex biomarkers that may need to be used. And there may be other things like imaging or other markers that could be used to help us to understand who are the right patients to receive these kinds of therapies, to avoid toxicities and to garner the benefit of these kinds of treatments. So I'm going to wrap up the presentation part on immunotherapy now and move on to precision medicine or targeted therapy in melanoma. As I mentioned, in thinking about the kinds of mutations that one sees in melanoma, they are centrally seen in the signaling through the RAS-MEK-ERK pathway. This is a simplistic depiction of signaling through that pathway. I'm going to try to do a little markup on this slide to help focus our thoughts. So the general outline of signaling in this kind of schema, as most of you would know, is that a signal emerges from a receptor tyrosine kinase shown at the top left. It impinges on RAS. There are multiple RAS isoforms, and that seems to be important in the biology. RAS then activates RAF, and there are multiple RAF isoforms as well. RAF can also be activated by mutation, as shown here with the V600E activated RAF protein. But the ultimate output of that is that RAF activates MEK via phosphorylation. MEK then activates ERK via phosphorylation, and then ERK, which is itself a kinase, goes on to activate by phosphorylation a series of downstream targets, which all turn on growth in the various ways in cells. Now, one thing I wanted to point out, which is important, is that NRAS is the most commonly mutationally activated form of RAS in melanoma. When you look at different genomic profiles in melanoma, there is a small rate of HRAS mutational activation that can be seen, unlike KRAS, which is pretty sparse in melanoma, but very commonly mutationally activated in a variety of other cancers. HRAS is interesting in that HRAS, is, like all of the other RAS proteins, needs to be attached to the plasma membrane via an isoprenoid tail, which is typically a farnesylated farnesyl tail that embeds RAS in the plasma membrane. There were drugs developed when I was a wee lass in pharma that target farnesyl transferase, but the biology turns out that only HRAS can be completely inhibited by farnesylation inhibition. The other RAS proteins can go on and be alternatively prenylated through granulation. So HRAS remains an interesting target, and some groups have gone on to try to move ahead the farnesyl transferase inhibitors that were developed at that time into more recent clinical trials. So I think that is an interesting kind of ultra-orphan situation in the case of melanoma, but something to think about for the future. But to really focus in now on what are the regulatory approvals in melanoma for various inhibitors in this pathway. They are vemurafenib, dabrafenib, the BRAF inhibitors, trametinib, and combination therapies of dabrafenib, trametinib, vemurafenib, and cobimetinib. There are other agents that are under clinical investigation, and some of them are shown here, including VTX11E, which is being developed now by Biomed Valley, I believe, and the shearing ERK inhibitor 772984. I'm not exactly sure of the status of that particular drug, but there was some early clinical development on that looked interesting. So interrupting signaling through this pathway seems like an obvious way to interfere with melanoma tumor growth. What one sees is that you need to do this in patients who have a V600 mutation. For vemurafenib and dabrafenib, that's V600E. The response in these patients is about half of patients experience a response, and those responses are dramatic. If you look at some of the images that are available in publications, in particular work by Keith Larry has shown patients with PET-positive tumors all around the body, and they just disappear. Dramatic lesions on the skin, they just disappear. But unfortunately, there are side effects, although generally manageable ones, which includes squamous cell carcinomas. But the real unfortunate part is that nearly all patients do experience progression and resistance in about a 1 to 2 year time frame. So there's been a lot of work underway to try to overcome this progression and resistance. And other agents have gone into development. For example, cobimetinib, which is also known as MEK162, is under review by the FDA for NRAS mutant melanoma, and we'll see how the review by the FDA proceeds on that. But there are other agents, as I mentioned, that are in clinical study. So that's just on the single agent front, and combination therapy seems like a way that one ought to be able to control the side effects and to potentially overcome some of the resistance. So the response rate with targeted therapy combinations is quite good at about 65%. Dabrafenib and trametinib, based on clinical studies, appear to be superior to vemurafenib as a single agent. Likewise, the combination is superior to dabrafenib alone, based on the clinical work that's been done. And Vem and Cobi in combination superior to Vem alone. Newer combinations are also being explored, including oncorafenib, a newer RAF inhibitor, in combination with vemurafenib. So a lot of work is proceeding in this area. I just want to show you a little bit of the clinical data to summarize and illustrate some of the points I made. This is a trial called data from the COMBI-V trial, which compared dabrafenib and trametinib in the blue line to vemurafenib in the orange line. And you can see that the combination, based on the overall survival data shown here, was significantly favored dabrafenib and trametinib. And if you look at a summary table that was published in 2015, which kind of pulls together all of the combination data on clinical studies that are being updated more or less as we speak at the ESMO meeting in Copenhagen that will take place in a few days, that these combination therapies do offer quite nice overall survival. Dabrafenib and trametinib with the longest history with a 25-month median OS shown at the top right. But Vem and Cobi, and we look forward to seeing what the data on encorafenib and cobimetinib is. These all appear to be superior to single-agent approaches. So at this point, I'm going to start to wrap up and talk a little bit about future directions. Would ask you to begin to think about questions you may have for me. But what about future directions? So the theme of the presentation was melanoma at the front lines. And what is the evidence for that? Well, the use of anti-PD-1 drugs, first approved in melanoma, has definitely been demonstrated in other cancers, in non-small cell lung cancer, in renal cell carcinoma, in lymphoma, and also in head and neck cancer. The PD-1 drugs that were first approved for melanoma are approved by the FDA for use in all of these other indications. There is very promising data for colorectal cancer that is mismatch repair deficient. And the hypothesis there is that in these particular cancers, there are many tumor neoantigens that are produced, and that these are fodder for the immune system to grab a hold of and to target. Likewise, in Merkel cell carcinoma, driven by Merkel cell polyomavirus, there is very promising data using anti-PD-1 drugs in this particular cancer. There are related drugs, I think of them as sister drugs, the PD-L1 drugs, right? Not targeting PD-1 itself, but targeting the ligand PD-L1. And there was an FDA approval for atezolizumab recently for its use in bladder cancer. There is promising data for another PD-L1 antibody called ipilimumab in Merkel cell carcinoma. So I think I and many others think that the biology of harnessing the immune system through PD-1 or PD-L1 targeting to disrupt the checkpoint blockade is going to have vast use in a lot of other cancers. And on the targeted therapy front, I think there was, my sense is that there was a little slowdown in enthusiasm for the use of these targeted agents when the responses waned and progression developed in such a large fraction of patients. But they are an important tool in the toolkit for melanoma, and they are gaining use in greater study in other indications. The BRAF-MEK inhibitor combo of dabrafenib and trametinib showed encouraging activity in BRAF mutant lung cancer in data that was presented in the Lancet quite recently. So my takeaways on all of that is that we should use all of the tools in our toolkit based on good, strong preclinical hypotheses and push these forward into the clinic. So just wrapping up, the development of immuno and targeted therapies for melanoma has been logical, it's been hypothesis-driven, and built on a deep biological knowledge, but most importantly, I have to say, on the commitment of some very important researchers who remained just laser-focused on the field and were supported by funders such as MRA to facilitate their work. And melanoma, I hope I've convinced you, as a case study not only for precision medicine, but also for immunotherapy, and that the lessons learned in this field are really being applied on the front lines for not just melanoma, but for many other cancers in the future. I'll stop there and open it up for questions.
Excellent presentation, Dr. Perkins. Thank you. Thank you for bringing that information to us. Before we get started with all of your questions, here's a quick reminder about how to submit your questions. You can send your questions via the green Q&A button at the lower left of the presentation window, and we'll get to as many as we can within the time allotted. Okay, let's see. I apologize, we're just having some minor technical difficulties. I appreciate your patience. Thank you for bearing with us. Our first question, Dr. Perkins, we have someone who would like some more information regarding combination treatments. What are combination treatments?
So the first question that I see is from Dr. Nasshoyer, what are the combination treatments? So the combination treatments in melanoma include dabrafenib and trametinib, that's approved, vemurafenib and cobimetinib, that is approved, and those are both targeted therapies. They depend on the presence of a mutation in V600. And if you take a look at the prescribing information, you can see much more detail in that. The other combination therapy that's approved for melanoma is that of ipilimumab and Nivolumab for the treatment of melanoma. So that's an immunotherapy combination.
Dr. Perkins, because we're dealing with a few technical difficulties, I know you can read on the screen, but if you can on the screen, the two questions that we had before, question number 1 and question number 2, if you could answer those, do you mind reading the question prior to answering it so our audience can hear the question?
Question number 1 was on the, I don't have the question open in front of me right now, but question number 1 was related to the biomarkers for melanoma. What are the prospects for biomarkers in melanoma? I think there's been some really interesting work that's been done to study PD-L1 as a biomarker, but there are so many opportunities in the biomarker space that need to be explored. There are biomarkers that are specific to individual treatments. The immunotherapy biomarkers that are being studied include things like immunoscore, the ratio of white blood cells in the circulation to white blood cells represented in the tumor. But I would also add that I think there is utility for a variety of biomarkers to be explored that might be broader on the basis of the disease itself, relating to depth of response or early relapse. And so these could be things like circulating biomarkers that are important to monitor the ability of any treatment to work and when to reintroduce new therapy early. So those are some of the kinds of things related to biomarkers that I think are really important. Other questions? I think we have some other questions coming in. Question from Dr. Martine Landrova. What about the interference that may be due to the presence of pathogens in patients, presumably like fungal infections or bacterial infections? I do not know. I do not know whether there have been any studies on this. I know there have been concerns about the use of immunotherapy agents in patients who have an autoimmune response. But I think that field is actually moving forward relatively well, and that exacerbating autoimmune responses has not proven to be as detrimental as one thought, but it certainly needs to be done with care. I think there was an early thought that the anti-PD-1 antibodies, based on their extremely safe side effect profile, were going to be easy to use, almost, I'll exaggerate, almost like aspirin. They do require some care and use to be able to monitor and manage side effects and to have patients pay close attention. Let's see. Any there's one other question that is available, and then perhaps we can start to wrap up. This question is also from Dr. Nashoyer. When will the drugs be available for lower risk melanoma patients? Most are approved only for the highest risk patients. Interferon is only marginally helpful but has a lot of side effects. Excellent question. So there's a lot of work that's going on right now to try to understand the use of, and I'll focus on the PD-1 agents, but it's also true for targeted therapies, the use of these drugs in the adjuvant setting as well as in the neoadjuvant setting. There are some studies that I know of that are being done with targeted therapy in the neoadjuvant setting. I know of one that's being done at least in part at MD Anderson for neoadjuvant and adjuvant therapy of PD-1 inhibitors. I think that work is also being explored. I would say that one concern that I've heard expressed is whether the combination therapy at the current dose for ipi and nivo, whether or any PD-1 in combination with any CTLA-4 inhibitor, but that's the approved one, whether that is appropriate to use in an adjuvant or neoadjuvant setting, given the side effects and the grade 3/4 AEs that are seen. But I think there's a great deal of interest in exploring the use of these agents in an earlier setting, as long as we're able to manage whatever side effects come about. And it's exactly in this setting that these early biomarkers that I was talking about would be particularly helpful, because the timelines to readouts are so long there. If we were able to build a data set that supported earlier readouts for these patients, it would be extremely helpful to advance the field.
I would like to once again thank Dr. Perkins for her presentation. Dr. Perkins, do you have any final comments for us?
Thank the organizers for the I just want to thank the organizers for the opportunity to present, and I hope you all agree with me that it's an incredibly exciting time, not only in melanoma, but in the rest of cancer. And for those of you who are researchers on the field, thank you for your efforts, and please keep up the good work. I'll stop there.
As a reminder, this webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Today's webcast will be available for on-demand viewing through February 2017. You will receive an email from us alerting you when it's available on demand and posted on LabRoots. You are welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging in and participating in today's broadcast. See you next time. Goodbye.